right, we are moving on to genetic technology today, and we are almost done with our units for the year. This is going to be like the last big one we cover. So we're going to talk about recombinant DNA technology. So the use of laboratory techniques to isolate them and manipulate fragments of DNA, so we can cut them apart, put them back together however we want. This process produces recombinant DNA, which contains DNA from 2 or more sources. Recombinant DNA can then be added to living cells, which will replicate the DNA, producing new copies in a process called gene cloning. And viruses do this a lot with DNA. You just don't think about people actually forcing it to happen. So in order to do it, we're going to start with step 1, which is a vector DNA and chromosomal DNA are starting materials to clone a gene. The vector is going to act as a carrier of the DNA segment that is to be cloned. Vectors carry only small parts of the chromosomes, or they carry single individual genes. Vectors come from 2 sources naturally. It's either going to be a plasmid from a bacteria, or they're going to be a viral vector. So plasmids are commonly found in bacteria and have been genetically engineered for us to use in cloning experiments, and we know that there's other good things that come from plasmids as well. We can use them to get rid of certain diseases if we want to. Of course, that is kind of still controversial today, in the United States at least. Viral vectors are from viruses, and they infect the living cell, and then they take control of its host. Step 2, we're going to cut the chromosomal and vector DNA into pieces, and then we're going to link them together to produce recombinant vectors. So we insert the chromosomal DNA into the plasmid or the viral vector. A restriction enzyme is then going to cut the DNA fragments for gene cloning. The sequences are going to be recognized by restriction enzymes, and these are going to be called the restriction site, and that's where we are going to end up cutting things apart. Certain restriction enzymes are useful in cloning because they digest DNA into fragments with sticky ends, and those sticky ends can then be stuck to other things. So they can be hydrogen bonded with other fragments, and we can just take those genes and insert them into wherever we want them to be. Step 3 is putting recombinant vectors into the host cells and allowing those cells to propagate to achieve a cloned gene. The goal is for the desired gene to be taken up by bacterial cells that have been treated with agents that render them permeable to DNA molecules, meaning they can go through DNA molecules. So DNA libraries can then be made in 2 different ways. We either have a genomic library, which is a collection of DNA from a single organism derived from chromosomal DNA. And this is from isolating mRNA using an enzyme to reverse transcriptase to make complementary DNA, and that's known as the cDNA library. The advantage of cDNA libraries is that they lack introns, and introns don't really code for anything, and it's kind of like junk DNA. They're just spliced in there, and they just hang out. They're normally very large, and then you have to rifle through everything to figure out exactly which gene you were looking for in the beginning. Most experiments are trying to clone particular genes. After making a DNA library, a researcher must isolate the gene of interest, and this is known as colony hybridization. This is where a researcher takes a probe complementary to the gene of interest to identify colonies that contain the desired gene. So here we have a master plate. This is all of the genes that they took out. And after they went through and did the hybridization, they selected just the genes that they were looking for and got rid of the rest. Southern blotting is going to detect the presence of a particular gene within a mixture of many chromosomal DNA fragments separated on a gel, which is gel electrophoresis. You may or may not have done an experiment like this in biology. They're normally very expensive, but you've probably read a gel electrophoresis chart. It's a separation of molecules using electricity. So they're put into this gel and they're charged, and then the DNA molecules will move from one end of the gel to the other end. The smaller fragments are going to move farther away, and this is used for DNA fingerprinting. So this is what a gel electrophoresis looks like. You would put all of the DNA into these wells up at the top, and these would be very long segments here, and you would be able to see them moving, like this little guy here moved all the way out here, so he's probably a smaller segment. The uses for gel electrophoresis, it's a lot of forensic science. So if you're taking that class, you probably studied the gel electrophoresis DNA fingerprinting. But for genetics purposes, you can identify different species of bacteria and fungi so that patients can be treated with the correct antibiotic. You can also do it for relationship testing. So you're determining maybe who the father is, or if they are a relative, some distant related cousin. You can also determine the identity of an individual if they have passed away, if they are in the system. I believe that if you are on any of those websites like Ancestry, they use DNA gel electrophoresis to track your DNA back to where your heritage is or where they believe it to be. Genomics is the molecular analysis of the entire genome of a species. So structural genomics is a direct analysis of the DNA. Functional genomics is the expression of a genome. Scientists are attempting to determine the roles of all cellular proteins to see how they interact to produce the characteristics of a particular cell types, as well as traits of an organism. In order to study genomes from entire eukaryotic organisms, we use a BAC, which can carry many more genes than a vector can. Once the DNA has been cloned, we have a map of the DNA and it's sequenced, which is a method to determine the base sequence. Now, a DNA microarray is going to be used to monitor the expression of thousands of genes all at the same time. So applications of microarrays, we have cell-specific gene expression. So we're comparing a microarray using the cDNAs derived from mRNA of different cell types. You can have gene regulation, which is environmental conditions playing a role on gene regulation being isolated from cells, and then they're exposed to two different conditions to see what exactly happens to that. For example, skin color, maybe you'll be a little bit darker if you're closer to the sun. Tumor profiling, so we're classifying tumors that are morphologically indistinguishable. So if they look the same, but maybe they act differently. Genetic variation, we're identifying disease-causing alleles in humans. Microbial strain of identification, we are looking at bacteria species. And I'm going to pause right there for a second and show you a quick video before we move on.
Researchers first remove samples from both the uninfected plant and the plant infected with the virus. mRNA is extracted from each of the plant samples. The mRNA is then reverse transcribed into more stable complementary DNA, called cDNA. The infected sample is fluorescently labeled red, while the uninfected sample is labeled green. At this point, in many microarray experiments, the two cDNA samples are combined. The combined sample of fluorescently labeled cDNA is applied to the microarray chip. The cDNA from the sample binds to its complementary sequence of DNA bases on the chip.
We know that the pieces that are fluorescently labeled will stick tightly to the corresponding gene features, but they won't stick tightly anywhere else. So we rinse the chip off after we do the hybridization step, and then we put the chip in a laser scanner to electronically capture the data.
The chip is scanned with a laser, activating the fluorescent dyes in the sampled cDNA. A computer captures this information, calculates the ratio of red and green on each spot, which indicates which genes are expressed in the uninfected and infected plant samples.
All right, so biotechnology, it began about 12,000 years ago, and it uses living things or products from living things to form things that are going to benefit humans, for example, food. In 1982, the FDA approved the sale of human insulin made by recombinant bacteria. Today, people use human insulin that was made by bacteria due to the development of certain allergies from using animal insulin. Bioremediation is the use of microorganisms to decrease pollutants in the environment. So we're using genetically modified things to help us. Enzymes are produced by microorganisms, modify toxic pollutants by altering the structure. We've been using this type of treatment in sewage plants along with industrial plants to break down heavy metals as well as pesticides. So the question that's currently being studied is, are these microorganisms that break down pollutants making them into something safe for the environment? So we're getting rid of these things because we personally can't do it ourselves, but what are they being made into? So here is the cleaning process. So we inject the microbes into the polluted water. So all these little spiky things are going to be the contaminants. The microbe is going to eat them or break them down, and it's going to be releasing CO2 and water. So carbon dioxide and water are pretty harmless, you would tend to think. I mean, we breathe out carbon dioxide, and as long as we have enough plants out there, they're going to be taking in that carbon dioxide and producing oxygen. So theoretically speaking, we should be all right. Genetic engineering. Transgenic is going to describe an organism that carries genes that were introduced using molecular techniques such as gene cloning. And here we have genetically modified organisms, GMO. These are transgenic things. Sometimes a cloned gene will recombine with a normal gene on a chromosome, known as gene replacement. In eukaryotes that are diploid, only one of the two copies is initially replaced. The initial gene replacement creates a heterozygote who will have one normal copy and one copy that has been cloned. If the cloned gene carries a mutation that inactivates its function, it is said to have undergone a gene knockout. So maybe we wanted to get rid of this normal gene because it's harmful to the environment or harmful to the organism. The function of the normal gene has been knocked out. We're using this technique when we study human diseases. So it'd be kind of cool if you do have something on a gene that's harming you, if we could go in there and basically inactivate it. We could potentially get rid of that disease. Cystic fibrosis in humans is caused by a defective gene. It has been identified, and researchers are using mice to experiment with various therapies in the treatment of the disease. We are trying to isolate the gene and then replace it with a normally functioning gene. Molecular farming is a production of medically important proteins in the mammary glands of livestock. Several human proteins have been successfully produced in the milk of domestic livestock, and we can treat certain strains of hemophilia and emphysema using this technique. Compared to the production of proteins in bacteria, an advantage to using livestock is that the proteins are more likely to function properly when they are expressed in mammals. Since we are mammals ourselves, if we can use another species of mammal, that would be better than using a bacteria because we're just closer, genetically speaking, to a mammal than we are to a bacteria. Animals can also produce more of the protein, which is then purified from the milk and given to the person in need. So now we're going to try to create genetically modified livestock, which we have done. I'll tell you the steps to that. First, you're going to insert the gene into a plasmid vector. You're going to inject the vector into the oocyte, which is an egg, where it will integrate into the genome. The egg is going to be fertilized by exposure to sperm, and then implants in the uterus of the sheep, for example, or a cow or a pig, but I'm going to use sheep. It should say sheep, not sheet. And then resulting in an offspring that will carry a gene and express it in the milk. So here we have the DNA that's been prepared in a lab. We're going to have this cow here. We're going to inject it into the egg of the cow. The egg will then be put into the cow and it's implanted. It's kind of like a surrogate. We can also make transgenic plants, and the process is much easier than in animals because plants are totipotent, meaning the entire organism can be made from somatic cells. We don't need sperm and egg cells. So a transgenic plant can be made by the introduction of cloned genes into the tissue of a leaf. Much simpler. You take the DNA, you put it into the plant cell. The plant is then going to grow and it'll produce babies, and then you get whatever you were trying to produce. Research with transgenic plants has led to the development of new products such as biodegradable plastics and medical products. Vaccines such as hepatitis B, cholera, and malaria are also from transgenic plants. Gene therapy is curing diseases using cloned genes, and it's kind of accepted, kind of not. More than 40,000 human genetic diseases involve a single gene abnormality. Even though large efforts have been made, few people have been successfully treated using gene therapy. Additionally, humans have had adverse reactions, and in 1999, a patient even died. Now, 1999 was, like 21 years ago? So I'm sure that since then we have gotten a little bit better at our gene therapy, but I'll show you a quick video, and that's where we're going to actually stop for today.
Many human diseases are caused by defective genes and improper protein functioning. Genes within our DNA provide the blueprint for the production of proteins. Specific gene defects or mutations can result in reduced or inability to produce a functional protein, or production of a protein with altered function. Gene transfer using adeno-associated virus, or AAV, vectors can target specific cells to correct disease-causing improper protein function. Once inside the target cell, therapeutic genes within the AAV can correct defective or missing protein function. To achieve this, AAV genetic material is replaced with a therapeutic gene to restore natural function of the disrupted proteins. A protein shell, called a capsid, encases the genetic material and helps target delivery to certain cells. Once within the target cells, the capsid is shed inside the nucleus to release its vector genome. The vector genome does not integrate into the nuclear DNA, but instead forms an episome. Episomes are not passed on to future generations of cells, thereby ensuring that the effect of the gene transfer is not passed on. Using the body's natural machinery, this allows stable and long-term expression of the therapeutic molecule. AAV-based vector delivery systems can be administered by intravenous drip or direct injection to target tissues. AAVs are ideal vectors for gene transfer, as they are non-pathogenic and capable of transducing both dividing and quiescent cells. The different components of AAV-based gene transfer can be switched to address different therapeutic targets and diseases, and several studies and clinical trials have indicated that there is activity in different disease models. Gene transfer offers the potential of long-term and potentially curative benefit to patients with genetic or acquired diseases by correcting defective or missing protein function through a single administration. uniQure is committed to developing innovative and cost-effective single-treatment gene therapies with potentially curative results.
All right, that is where we're stopping for today. Have a good one, guys.